Okay, well, welcome to Iowa Pork Industry Center's regular webinar series. My name is Colin Johnson. I'm one of the swine field extension specialists for Iowa State University Extension and affiliated with the Iowa Pork Industry Center, which has the pleasure of bringing you these webinars. And today, I'd like to introduce Dr. Juan Steibel and look forward to hearing from his insights and experiences in precision swine management and just precision tools. But Juan is a native of Argentina, where he obtained a bachelor's in agronomy and a master's in animal breeding and biometrics. After that, he went to Michigan State University, where he received his PhD in animal science. Juan was on the faculty at Michigan State until 2022, when he accepted the current position here at Iowa State University as the J. Lush Endowed Chair of Animal Breeding and Genetics. Here at ISU, Juan's research focuses on phenomics and genomics of animal agricultural species, especially of pigs. And his main interests are on the genetics of behavior and its application in the selection of animals better adapted to better group housing conditions. Juan's research also delves into the utilization of sensing technologies for management, breeding, and research in pigs, which is today's topic of the seminar. So welcome, Juan. We'll look forward to hearing your insights, and I'll be paying attention to any chat items and time for questions as well. So welcome.
Excellent. Thank you very much for the introduction, Colin. Let me share my screen. Okay, and put a timer on so I don't digress. Thank you for this invitation from the Iowa Pork Industry Center to present. Even though I don't have an extension appointment, most of my major appointment is in research. I think it's very important that we, as part of a land-grant university, we all engage in these outreach activities, because it's the way in which we can have an impact in the real world. So having said that, and because I am mostly a researcher, and my research is mostly on using automatic phenotyping for studying the genetics of behavior. Today we'll talk about a lot about that. But I will start with a brief introduction of what is PLF. Then I will talk about precisely using data, behavioral data in genetics, and then how PLF sensors might play a role on that. And then I will talk about what are the new projects that we are starting at, or just started at Iowa State with collaborators. And I will close with some ideas of how PLF data can be used for many other things, and maybe uses that are already incipient state, but that we should all pay attention to, because this will develop very quickly. So let's start with what is PLF. There's several definitions. It is an emerging field. It's well established now, but it's a new field within livestock farming in general. And it's one of the definitions is that is the application of technology in the animal space to perform automatic real-time monitoring of individual animals. Of course, this is quite restrictive definition. Then it can be relaxed, and instead of working with individual animals, working with small groups, etc. But the idea is the key words here are automation, real-time, and hopefully individual animal as well. And then this idea of PLF, or the field of PLF, hinges around a conceptual term or conceptual construct that was coined by Dr. Daniel Bergmans, who's been now informally credited as the godfather of PLF. And we pretty much all agree that to work under this premise, that animals are complex, individually different, time-varying, and dynamic systems. And as you can imagine, this is quite a engineering-based definition. It's not surprising that an engineer came up with this, but it's very handy for organizing the way in which we work and we think about PLF, and in general sensing for PLF. The first one is that the animal is a complex system, and a complex system, if we want to monitor it, we need to record many variables that for us geneticists and maybe for other animal scientists as well are called traits or phenotypes. So we need to look at several of those simultaneously. The other premise is that animals are individually different. So if we want to tailor management, we need animal identification. We need to individualize animals or at least narrow it down to a small number of animals. Then in some other cases, technology can be used to improve the management of the group. And in that case, the group has to be identified. And that's what is feasible sometimes, but in the long term and the dream is animal identification for individual decisions. Then the other two are related, but they are different from each other. Is that these animals are time varying and dynamic. By time varying, it refers to the average or the mean trajectory of each of the traits which are important to monitor over the lifetime of an individual. And so that baseline that before I said that is individual baseline. Now it's also a baseline that changes over time as the individual grows, develops, and changes in its physiological states. But even so imagine like a time average trajectory for each individual. But it's quote unquote an average trajectory because the dynamic nature of it involves like fluctuations around that expected trajectory for each individual. And precisely monitoring those fluctuations is what makes possible, for instance, to detect problems with welfare, health issues, etc., that require intervention. So these are the four premises, and from time to time in my presentation, I will refer to some of them. So let's start with what is not new here, and it's the concept is the management of a production system. It could be anything that you can think of. To do that, we define some key indicators. Could be, I don't know, body condition score, some reproductive performance traits, feed intake, whatnot. And based on that, we decide which variables we need to measure or monitor. And we need to measure as many as possible to make informed decisions. And we've been doing this in animal management for years, decades, and I would argue even centuries. So based on this, these measurements, we try to reconstruct the key management indicators, and based on those, we take action. This is nothing new. I think all of us, whether we manage any type of system, we have to use this. But what is kind of new with PLF? The idea with PLF is now to monitor these variables or these traits. We introduce sensors in the proximity of the animal, and with those sensors produce an enormous amount of raw data that hopefully the person doing the management never sees. if we have to look into that, probably you are in trouble already. And those raw data are processed into usable data. And that usable data or management flags, if you want to call them that way, are used by the animal caretaker to take action. And then after that, necessarily, the data has to be discarded because there's no way to store all this. Even if you try at the beginning in any real life application, it will very quickly mushroom into an unmanageable amount of data. Now, for research purposes and for research and development at public and private institutions, though, we store as much as possible of the raw data, of the management flags, etc., to make use of this, and we'll talk about this briefly later. So there are many types of sensors. This talk is not completely sensor agnostic, but I don't want to dismiss other type of sensors that I might not mention today in the application that I showed. But I tend to classify these sensors into two large groups, depending on how they position with respect to the animal that we are monitoring, or the animals. One type are the remote sensors, and these sensors are, as you guessed it, away from the animal. In the case of a pig barn, it can be in the ceiling, or it can be on the wall. It could be a camera or a microphone. In the case of free range production conditions. That could be like in a cow-calf operation, for instance. There might be as remote as in a satellite or in a drone that is hovering over the pasture, etc. But the idea is that it's not attached to the animal. That's the difference with the wearable sensors that many of you have heard of, and there's quite a few in dairy cattle and in even for beef cattle. There are some now. There are less of those, but there's one that is very commonly used in some uh, pig production systems or even in nucleus farms, which is the RFID. RFID. So these are attached to the animal, and of course, to collect the data, there has to be some sort of collection device that is away from the animal. It could be an antenna, a beacon of some sort. And then there's what I call the proximal sensors, and the proximal sensors are sensors that the animal approaches, and as the animal approaches, start to record some data. Most of the PLF and phenomics technologies combine sensors. For instance, combine RFID with a load cell in an automatic feeder to measure how much feed was eaten by a finishing pig in a nucleus farm, for instance, for selection purposes, to estimate feed intake or residual feed intake. So today I will talk mostly about camera technologies, and I will talk about some of these devices using automatic feeders. You might or might not know this, so if it's the moment to sneeze or look away, you can. Won't miss much. But anyways, for completeness, when we put a sensor on the animal space, the sensor produces, as I said, an enormous amount of raw data. The raw data is almost unintelligible for a human, and that raw data is analyzed with something called machine learning model. Is basically a computer model, a piece of computer software that gets that data plus some known characteristics of that data, and it produces a quote unquote a raw phenotype. For instance, in this example that you have here is a model that is being trained to look at images and then draw a silhouette, which is called a mask, around the dog and the ewes in a sheep operation. This other model is being trained to look at image of a horse and place key points on very important joints or body parts that are used later. So this first model is just learns that from an image, it learns to obtain these raw phenotypes. And hopefully that works, and I will show some examples where it does and where it doesn't. And then after that, after we have these raw phenotypes, there is one more machine learning or another computer piece of computer software that will transform this raw phenotype into what I call operable phenotypes, operable traits, management flags, or distill phenotypes, which is basically will tell you something about what is going on there that you can work on. For instance, it could transform all the key points into a locomotion score. It could transform the mask and other characteristics of the mask into an estimate of the body weight of the individual that's being looked at, and so on. So what we care about this only at the end of the day, and we want these to be accurate and representative of the real life scenario that is being monitored with the sensor. But to get to this, there's many steps, and so it's usually good to know conceptually what these steps are, to understand where this can fail, and to understand how we need to evaluate this to make sure that it works. So I said before, we measure variables in a system to get to some key indicators or key quantities for management, to inform management or selection or whatever we want to do, whatever intervention. And there was nothing new in that. What is new in incorporating PLF sensors and sensors in general into these is that it's new in two aspects. The one source of novelty is we can look at now at new traits. And these are traits that before we were not able to easily monitor in our production systems. And I mentioned some of these, but I'm sure you can come up with more. For instance, one that I'm very interested in is monitoring behavior, because through looking at behavior, as we will see, we can do many things in genetics and in management. Another source of novelty is that maybe we are collecting sometimes the same traits that we collected before, for instance, individual body weights. But now we can collect them real time, almost continuously, hopefully continuously, in many, many animals across farms worldwide, and then summarize them or collate them into a single place, and that has a lot of value. Accumulating that type of data has a lot of value. And what is more important is that a lot of these can be done now, or hopefully we want to do it under normal production conditions. And while before, these sensors maybe were constrained to be used in research farms or in nucleus farms, let's say, in data collection facilities that were not the actual production environment, necessarily the actual production environment. So that's a brief introduction of what I'm talking about when I talk about PLF and sensors, etc., and the context in which I tend to evaluate and look at this. So now I want to share with you some of my work, and the work I've done with, it's not just mine, it's also with collaborators, on the genetics of pig behavior and how it connects to automatic phenotyping and sensing. So the one very interesting one that actually got me into PLF is this idea of trying to observe behavioral interactions in pigs specifically. And more specifically, I worked a lot on growing pigs, but my ultimate goal on this is to also work on gestating sows, because as you know, there is a increased emphasis on group sow housing during gestation. And of course, I'm interested in the management and selection of sows that are better adapted to group housing, something that we didn't have to deal with 10 years ago at all in the US. So, from the point of view of breeding and genetics, I'm interested in behavior and measuring behavioral interactions for two reasons. One, there is without doubt, there is an effect of the genetic makeup of an individual on their behavior. And through that, if we see some undesirable behavioral interactions, there is always the possibility of selecting against those. If we can observe them, we cannot, if we cannot phenotype, we cannot implement breeding value prediction and selection. That's one. So looking at the behavior as the actual end trait. And the other one is that through looking at the behavior, we can select animals that are better suited for group housing, because, for instance, we can select animals that compete less, or animals that affect less the performance of other individuals in the group. And that's called an indirect genetic effect or social genetic effect. It's an inheritable effect of an individual that is not expressed in its own performance, but is expressed in the performance of all the social group mates or all the animals that interacted with that individual. And that connection between this DNA and this phenotype here has to be through behavioral interactions. So I'm interested on this for these two reasons. And this started a while ago with a collaboration between three institutions: Michigan State University, and here's Jenny Sigford, Scotland Royal University College, Simon Turner, and University of Buenos Aires, Fito Cantet and Belsi and Garita. And basically what we did back then is we had deeply phenotyped individuals for lesion counts in the skin post-mixing, and also for their behavior. And all this phenotyping was done, quote unquote, manually, without any sensor. So basically, there was a camera recording the behavior of the animal. There were also trained observers counting lesions on about 1,000 pigs, and this counting was done right after mixing in the finisher. As you know, you are very familiar with pig farming. After mixing, there is intense fighting until the animals reach a stable social hierarchy, and the fighting diminishes. It doesn't go away completely, but it diminishes substantially. So we were looking at those 24 hours post-mixing. We look at this steady state, but I won't present those results. And so what we did was use the behavioral interactions that were recorded and decoded as a way to, together with the DNA data, genomic data from this individual, as a way to study the lesion counts as a phenotype. And what we found is that by incorporating the behavioral observations into the modeling of the lesion counts, we can recover much more variance, and we can not just the direct genetic variance, but we could also recover the social variance. So if someone was to select for animals that showed less lesion counts, and it would be very effective to incorporate the behavioral interactions. And it's not selecting for any behavior in particular, it's just selecting for animal that causes less lesions in the group mates. Without that data, most of the work is restricted to understanding the lesion as an effect of the victim. Maybe there is some proxies about who can cause the lesion based on the distribution, but that's another story. Now, once we start to put behavioral interactions, we can select animals that tend to have progeny that causes more or less lesions. That's the end of the story, or the moral of the story. But there was one problem. This was all done manually. So I thought, well, I was reading a lot about sensors and cameras and those sort of things back then. So I said, well, this is great for applying computer vision and trying to extract some automatic phenotypes. So I will get to that later. Another thing that we started to look at is with a different type of sensor with automatic feeders. And these automatic feeders are used a lot in nucleus farms to collect data for selecting animals for feed intake, for more efficient, that make more efficient use of feed. And basically the data comes in a way like this. This is just I open an example data set in Excel from an old experiment at Michigan State. And there is one line for each visit. There is one line for each visit to the feeder. And then there is for each line, there is a RFID. So we know the ID of the animal because it reads, the antenna reads the RFID that is in the ear of the pig that is eating. And then there is an entry time and an exit time and the amount of feed consumed, etc. And the way that these data are usually used is you sum all the visits from one animal, and then you sum the feed intake, you sum the time of the feeder, and then you have an idea of how much the animal ate over a period of time. You also have weight again, so you can estimate feed efficiency and whatnot. And our idea, or my suggestion, was let's look at the actual sequence. Who comes after whom and what happens with the animal that is eating when one animal comes behind it. And for that, we look not only at the time that an animal stayed at the feeder, but also at the distance in time between two consecutive visits. And the idea was that when the animals were replacing one after the other quickly, there is a potential for having a lot of competition for feed, while the replacement rate is slower and there's more time between two visits. We assume that there is no competition. So then we started to see in these two contexts, hypothetically no competition or hypothetically intense competition, if the ID or the identity of the animal following a feeding animal affects the amount of feed and the time that this animal stays at the feeder. And we published this also way back, but now here at Iowa State, we started a collaboration with PIC, and through data sharing agreement and my student Andrea Nunez, we started to look at these in a very large data set. And the actual model doesn't matter today, but what matters is that in this model we have a genetic component and an environmental component, environmental component for both, for the direct effect of the animal that is eating and for the indirect effect of the animal that is following. And this only is possible because all these animals have RFIDs, and there's an automatic feeder that measures how much the animal ate, how long it stayed at the feeder, how long after an animal left another one came in. So without talking about any specific results, just roughly speaking, what we found is that when there is a long time of replacement, there is very little variance, if any, explained by the peak that follows, and then in the variance in the meal length of the animal that is eating. And when there is a quick replacement, or those times when the feeder is used intensively, the animal following matters, and it can imply some variance in the amount of time that the feeding pig stays at the feeder. And this is very interesting because if it is confirmed, it means that someone could use this data to select animals that compete less for feeding space. And this is my, of course, my pride as a geneticist. Remember, any genetic solution that we implement is permanent, and management solutions have to be implemented always. This is just a geneticist joke that we usually make. So this is promising, I guess. So all these, so far, in all these results that I presented, there is no automated observation of behavior. There was just manual observation of behavior, or using a proxy, which is just the sequence of visits. We don't know what the animals are doing at the feeder, for trying to find animals that are genetically better for either sharing feed, or not competing so much for feed, or not attacking each other that much after mixing. So can we automate the recording of behavior? And back then, several years ago, I thought, well, this is great. Now with the automatic phenotyping, I will solve my problems. Well, it didn't solve my problem, but it creates some more, which is good for a researcher because problems mean new things to work on. So one thing that we started with my former student, Junjie Han, was to look at exactly what was going on in those feeders. And so we put a camera on top of the feeder, and we started to look at what the animal, not the animal that is eating, like in this case, but what the animal that follows is doing. And so our first attempt was to classify, looking at short videos, to classify what the animals were doing. So in this case, there were two pigs, but basically the following pig was doing nothing. In this one, is just maybe exploring the other pig. It's not a very intense interaction. Then there was more intense ones like this, livering and mounting. So can we detect these interactions automatically? And so again, there is a very complex machine learning, and the details don't matter, but people who do this, like Junjie and my collaborators, a lot of people in my group know a lot about this, and they prepare these machines. And so what we do is we look at the accuracy. So we have some data where we observe manually by just a human observer. It labels the data. We process it through a computer. It's data that the computer never saw before. So we look at the errors, and basically let's look at the precision with which these 4 different types of interactions were called. And they were very, very nice. I mean, the precision was around 0.9 to 0.95, which is excellent. But then let's look at different social groups. So let's train on some individuals and use other individuals for prediction, which is a more realistic scenario. And in that case, the performance suffer a lot. Now, this is not always the case. This was one set of a large set of studies with animals of different time points in the finishing cycle, different animal groups of animals, etc. But we observe this. So this is something to consider. When we validate these technologies, we have to look at the validation over a wide range of conditions, not just within one data set. No matter how big or large or small the data set is, if it's just one data set, we might run into the problem of being overconfident, believing that it's very precise, but then as soon as we look at new data, the performance might suffer. And then with the same idea, my student here at Iowa State, Mina Shumali, started to look at other ways to look at this and detect the peaks and try to focus on the area that matters to see the interaction, etc. And again, depending on the week, the date, etc., these models might work not so well. So validating this is very important. So far, what we found is that these models are very effective in detecting feeder use, detecting very moments at which there is potential for competition because of a lot of visits. So now, for instance, we could have an idea of competition at a feeder without having to have all the other measuring devices, but for specific interactions at the feeder, we are not quite there yet with our computer vision. Maybe other groups are, and but it's certainly if they are, it's because they have more data and they have a wider variety of data. The problem is the broad application of this. So with that, with just that examples, I want to go into some new initiatives that we have at ISU, and I don't have a lot of distilled results yet for this, but some promising ones. One is a collaboration with Santosh Pandey from computer engineering, and with two of my students, Mina Shumali and Will Taylor, and these two complement each other very well because Mina is a expert in computer science and algorithms, etc. And Will is an expert in animal evaluations. He's the coach of our judging team in the department. So we look at the way that Will evaluates animals visually, and we try to emulate that with the computer. So anyways, the idea is the first run around was let's use a model that already existed and was trained elsewhere to try to detect key points in these pigs. And sometimes it worked, sometimes it didn't work, as you can see. So we set off to train our own model for this. And the idea is this research is done in our swine nutrition farm here at ISU, and using very simple, this is not specialized cameras or anything, it's a very simple recording device, which has its shortcomings, but it has one advantage, it's very popular. So if we can make it work for this, it will work in many different other applications. And again, following the work from Will and what Will looks at in the animals, we decided on which key points we'll monitor, and then we collected some data and pigs of different ages. We are very interested in this case in young pigs. So if we can, for instance, through looking at locomotion analysis, we can detect some issues, for instance, with nutrition, or even evaluate feet and leg very early in the life of the animal. And once we train a model, it seems to work much better now. This is a result of the model applied on an image that the computer never saw before, on size of pig that never saw before. And in this case, what is interesting for this type of models, we have very high accuracy, even if we train in completely different contexts. So that's very promising. And another thing that we are looking at is, because we have all these morphometrics, and now we can use some of those for A, identifying animals, B, estimating body size, and of course, as I said, the classical use of this type of output, which is the locomotion and feet and leg score. So at this point, this is where we are right now. At this point, we are in that first model that I showed you before in the slide with the machine learning. We can extract the key points, and we can kind of make sense of those. Now we are working on the last model, which is to study locomotion in these animals. And another one that is being led by David Rosero, that many of you know well, is dedicated to the monitoring of pigs in the nursery, monitor feeder use and monitor drinker use, and not feed intake. Maybe this, I should change this, feeder use and drinker use. And the idea of this is that if we can identify animals and look at the use of feed and water, especially early, as soon as the animal gets into the, as soon as the pigs get into the nursery, we might be able to detect problems early. Like, for instance, if there is a diarrhea coming up, it should be detected at the, even at the pen level without animal ID, by a change in activity. The other one is adding the animal ID, we would be able to detect what are the behaviors or what are the resource use of those quote unquote non-starting pigs. So pigs that struggle to get off the ground and then perhaps they carry on. There might be some compensatory growth, but they carry on that issue throughout their whole finishing cycle. So as I said, this is something that we are working together, David and I, and his student is leading the data analysis of this. There will be more coming up. So through all this, I think I need to wrap up, but what are the challenges that I see for this? One challenge is in this, with this, coming back to this slide that I showed before, one big challenge is to validate in broader context. Even for commercially available technologies that tend to work quite well, one thing that we are observing is that they require sometimes quote unquote fine tuning or adding more data or refining these machines for better prediction. Of course, those early adopters of technology get all the attention of the companies, and then they contribute a lot of data, and that's the way in which the company improve by delivering, by selling products, and then improving on the fly. I call it the Microsoft Office model, which release a new version, wait for some crashes, report from the users, and fix them quickly that day. So this is kind of one way it's working, but we need to think very deeply about this. One very interesting initiative that is going on in our department is we have a project with David Rosero again to create a test bed, a farm that could be a test bed for technology under broader context, and a farm network. So that's really exciting. The other one that I am extremely interested in as a breeder is this problem of animal identification. Of course, there are several solutions already available for this, but they are not quite widely available maybe, but there are already some. But one problem that we have is once we extract the phenotype, how to link it to an animal ID that is compatible with all the records that we have on farm. For a breeder could be pedigree, genotyping records. For a manager could be other key performance indicators, previous life history of the animal, and even health treatments and management interventions done before. So this is something that is very important, is at the core of PLF also, and also at the core of phenotyping for breeding, as I said, because we need animal ID. And of course, there are many methods for identification, but ones that seem to be now in use is a combination of approaches. For instance, we know that PIC is already pushing the field on this, and they basically combine short-term tracking with visual identification of the animal, so through a proprietary set of tags that are very cleverly designed. Other like research applications recently published, they combine short-term tracking with RFID at the point of contact, in this case at the feeder. Very interesting work from University of Wisconsin. And also there is quite a bit of work on morphometrics and using some of these animal dimensions that can be extracted from key points to try to come up with an animal ID. This is very common in facial recognition. It's used a lot. It's just facial recognition is not very useful in livestock farming, whether it's a dairy cow or a pig. Usually cameras are on the ceiling, looking down, not straight down, maybe angled down. And so the facial features are not recognizable, but the same principle perhaps could be applied to other animal dimensions. And this work from University of Wisconsin in dairy cattle, and this work from our lab is currently being produced with pigs. It's just limited to the number of animals that can be distinguished. If there's 10 animals, it's easier to detect based on to identify based on morphometrics. If there is 100 animals, it's harder. Okay, and we are getting close. So just two promising uses as we go, as we implement more and more PLF in farms, we'll be able to collect a lot of data, and those data sets will be used for comparing products. And now the experimental unit is not the animal or a small pen, but each experimental unit is a whole barn or a whole farm. And the output maybe is just economic output from that. So this is very exciting. I don't have a lot, just these to mention. I think this company is already trying to mine this data. That's why it's very important to have a conversation about who owns the data, how the data is used, because there is a lot of potential on this area. The other one that I will skip is for individual welfare assessment. So I want to close with my reflections of what we need to advance this field more from the research point of view, let's say in a land-grant institution. We need properly validated and interoperable PLF. Interoperable because data from sensors can be used for more than one purpose. And that, in my experience, involves the IT piece and all the data analysis, transmission, storage, etc. That may be an issue in some locations. We need to think about data ownership, data usage, data sharing. But what is most important at the base of this pyramid is the interdisciplinary work, and not just in research, but also the interdisciplinary training of the new animal scientists, of the new engineers that will have to work together in the future. Anyways, I think with that, I will finish my presentation and I will open for any question if we have time.
Thank you, Dr. Steibel. I'm checking the chat. We don't have any at the moment. If anybody has a quick chat opportunity, go for that. And we'll remind attendees that look forward to kind of some further research from Dr. Steibel coming forward, obviously, and appreciate that. But I'll remind you that these webinars, if you happen to be just tuning in and listening, they are recorded. And there's a whole variety of those recorded if you want to share with colleagues. Also, if you go to the Iowa Pork Industry Center's website, we have a vast library of past recorded webinars and more upcoming. But the Pork Industry Center website, it would be ipic.iastate.edu, and you can find a whole variety of those webinars on industry issues. So not seeing much for chat, but on behalf of the industry, Juan, I do want to thank you for your work in this space. Certainly look forward to a lot of upcoming research and obviously further advancements from that, both in regards to production efficiencies and well-being measures as well. So with that, I bid you a great day and appreciate it, Juan.